Immunofluorescence is a microscopy-based technique that detects specific antigens, which are proteins or other cellular components, by using fluorescently labeled antibodies. For the principle, we have fluorescent antibodies bind directly or indirectly to target antigens within a sample. When exposed to a specific wavelength of light, the fluorophore emits light, allowing visualization under a fluorescent microscope. So how it works is we have antibody binding. So the primary antibodies target specific antigens or proteins within the cell or tissue. The antibodies are conjugated with fluorescent dyes. The microscopy, so after binding, the sample is examined under a fluorescence microscope, which uses a light source, either usually UV or blue light, and filters to visualize the emitted fluorescence. For the detection, the antigen's location and quantity can be determined by the color and intensity of the signal. There's different types of immunofluorescence. We have direct immunofluorescence. This is where a labeled antibody binds directly to the antigen. This is faster and has less background noise. And then you have indirect immunofluorescence. This uses an unlabeled primary antibody and a fluorescent secondary antibody. This is more sensitive due to signal amplification. These can be used in autoimmune disease diagnosis, so it can be used as an ANA or an anti-nuclear antibody testing for systemic lupus. Can be used for anti-basement membrane antibodies for Goodpasture syndrome, also for anti-skin antibodies for bullous skin disorders. Can also be used for tissue-based detection for kidney, skin, or lung biopsies to visualize immune complex depositions. Can also be used in microbiology for identification of bacterial, viral, or fungal antigens in patient samples. Some things to remember for immunofluorescence is it provides highly specific localization of antigens in tissues or cells. It is crucial in the diagnosis of autoimmune, infectious, and dermatologic diseases. Its results depend on sample quality, antibody specificity, and microscopy techniques.